Important to know. I ask my students, what are the two phases of the cell cycle? And they say prophase, metaphase. No, those are the phases of mitosis. So the cell cycle has two phases. The brown part, the small part, mitosis, and the very big green part known as interphase. That's very important to know. The cell cycle is made up of mitosis and interphase. Now, each one is divided into separate phases. How many phases is interphase broken down into? Three phases. You need to know those. The G1 phase, that's when the chromosome, the cells grow and they adjust their normal metabolic rate. And the S phase is when the DNA is replicated. You have doubling the DNA to prepare for division. And lastly, you have the screening of this DNA that we did not double up a DNA that is damaged. We don't want to double a DNA that is mutated. So we screen the DNA, we grow, we prepare for mitosis. Then we enter the mitotic phase. You will see that the same way, each phase of interphase had the distinctive feature. So you need to know what are the distinctive features of G1, S, and G2. G1's distinctive feature is that it's the phase of the growth and the normal metabolic functions. The S phase is the phase where there is the DNA replication. And lastly, you have the second gap phase, and that is the growth and the preparation for mitosis. Then once you enter mitosis, you have prophase, metaphase, anaphase, telophase, and cytokinesis. You need to know the distinctive features of each of these phases in mitosis. Prophase is known for what? Is known for the fragmentation and the breakage of the nuclear envelope, the nuclear membranes, known as karyorrhexis. Karyorrhexis means we destroy the nuclear membrane. In fact, we need to do that because how are we going to divide the DNA and the nuclear membrane is still there? We need to remove the nuclear DNA. Then in metaphase, we line up the chromosomes at something known as the metaphase plate. What's the metaphase plate? The metaphase plate is an arrangement where we line up all of the X's, the chromosomes, at the middle of the cell. Then anaphase is the most easiest one to identify because you see that the plucking off. You see these two sister chromatids came apart because the microtubules attach to the kinetochores and they are pulling in opposite directions. Telophase is the appearance of the cleavage furrow and